Dehydration builds gains. Hydrolysis? Bro, is just catabolic. Let's hit AP Bio 1.3, hun. But yeah, note the disclaimer.
This summary table basically will provide you a brief overview of understanding the similarities and differences between dehydration synthesis and hydrolysis. So when we talk about dehydration synthesis and hydrolysis, please note that we are talking about this idea of either breaking or building bonds. So in dehydration synthesis, we are removing water, while in hydrolysis, we are adding water. And the goal is to eventually create macromolecules. Macromolecules are considered polymers or connections of what we call monomers. And if you remember, the different types of macromolecules include things like carbs, lipids, proteins, DNA. So both processes work hand in hand to either create macromolecules or break macromolecules. Now, in this section 1.3, we're not focusing in on the specific macromolecules, and this has changed from previous years because the focus is just on the process listed here. In later sections, you'll focus on more specific details about how dehydration synthesis and hydrolysis is utilized to create and break bonds with these different macromolecules. So I just want to note that as a previous disclaimer. Please note that for dehydration synthesis, you're joining those monomers. So basically you have these connections, you're joining them together, and they form these polymers, and that is done by removing the water in the process of dehydration synthesis. You're essentially removing from one monomer a hydrogen and another molecule, or I should say monomer, a OH, and that OH is also known as a hydroxyl. So if you notice here, there are two H's and one O. So essentially, you're removing one water molecule, which allows for the formation of a bond. This requires energy, sometimes in the form of ATP, and is requiring enzymes. This allows us to build those macromolecules, whether it be carbohydrates, proteins, lipids, or DNA or RNA, those nucleic acids. This is an example of an equation in which you have these two monomers, they're joining together, and they break off a water molecule to create even a bigger molecule. The goal is to build these macromolecules or these polymers by removal of water for dehydration synthesis. Now, for hydrolysis, you'll notice we're adding water, and the goal is to split those polymers, or there's multiple connections, basically by breaking them apart into two or many separate little parts, which are known as monomers. The purpose is to add water, and then basically that H2O is split, where now you have the H and the hydroxyl. So it splits the water molecule into one H for one of the H2O, the one of the hydrogens in H2O, and the OH is from the O and the H left over. So essentially it's breaking that apart, breaking the covalent bond, therefore releasing that energy. Yes, there are enzymes that are required as well, and you'll notice the similarity between both dehydration and hydrolysis with these two functions of dehydration synthesis and hydrolysis. The purpose of hydrolysis is to allow for the splitting for processes like digestion, metabolism, molecule recycling. So metabolism, digestion, and molecule recycling are just a few of the processes that use hydrolysis. And the goal, once again, is to take that water and have that polymer and break it down into their monomers, essentially, breaking down using water. So now you can see the similarities and differences between dehydration and hydrolysis. If you ever need a little hint, hydrolysis means water death. If you think a Lysol wipe, you think Lysol or death. So we're adding hydro, water, to then kill or split apart a polymer. That's the way I think of it. And then dehydration synthesis, you want to think about it if you're hydrating yourself, you're giving yourself water. But if you're dehydrated, that means you have removed the water. The water is away from you. So dehydration synthesis. This is a great summary table, and now we're going to focus in on how the actions and mechanisms actually occur.
Hey guys, you want to hear the tea from your favorite water molecule, H2O? I'm just built a little different due to dehydration synthesis, and here's the tea about how it went down. I wasn't even born yet, and these two monomers were chillin', and they decided to link up. But to do that, they had to sacrifice a part of themselves to make room for this bond. Monomer 1 was like, yo, I'll drop off a hydrogen, and monomer 2 is like, cool, I'll lose a hydroxyl group, or OH. And then boom, there was me, H2O, on the run. Talk about drama. They bonded, I bounced. That's dehydration synthesis. Literally removing water to build something bitter. These two monomers joined together to basically form what's called a polymer, because I was removed and now I'm on the run. But basically, this helps create things like carbs, proteins, nucleic acids, and lipids. Sometimes I require energy or ATP, but building macromolecules is a must due to me being removed at a molecule breakup. That's the tea.
Let's take a look at the term dehydration synthesis, which is the opposite of hydrolysis. The term dehydration basically means removal of water. So if you're walking around and someone says you're dehydrated, that basically means you don't have water. You've removed the water. And synthesis means to build. So we're removing the water to essentially create a polymer or to build a polymer. So the goal of dehydration synthesis is essentially to take a monomer, more than one. So we have one monomer here. So we have this whole monomer here and this monomer here that we see in this picture. And we're trying to combine them together. We're trying to link them together. The way that we do that is we take the hydrogen from one of the molecules or the monomers and take the OH from the other monomer, and we go ahead and create water, which is removed, essentially creating this breakage point between both of the monomers. Those are joined together and they create a polymer to build a new bond. This is great, especially if we're trying to build from sugars, things like carbs, proteins, or nucleic acid. And this is great because it allows for the assistance of creating those items. For example, if we took glucose and fructose and we combine them together, we would make sucrose plus water. So the idea is to take our hydrogen ions and our OH ions, basically hydroxyl, combine them together to remove to form water. This process is known as polymerization.
Yo, it's me, H2O again, the real MVP when it comes to breaking up stuff. Yeah, we talked about dehydration synthesis in the past, but here I'm hydrolysis. The H2O breaking it down, literally. See, this big polymer over there, yeah, I pull up and add myself to the mix and suddenly, boom, all of a sudden there's two monomers now. Why? 'Cause y'all need to digest your food, recycle molecules, and keep your body running. I help release energy this time around and drop off some fresh building blocks for your cells. No big deal. Just saving your life molecule by molecule. I'm the bond breaker this time around. Hydrolysis hero. Just built a little different, y'all.
Let's talk about hydrolysis in more detail. You can see here the term hydrolysis, and you can see the root word here, hydro and lysis. And when we put that together, that means water split. So essentially, hydro meaning water and lysis means death. We're taking this polymer and breaking it down into these monomers over here. The way that we go ahead and do that is by adding water. And as you can see here, there are two hydrogens for every one oxygen. And the reason why this is important for our whole structure is that we're taking this polymer here and we're saying that there is a covalent bond somewhere in that structure. By adding our water in, these bonds are now broken, essentially producing smaller molecules where they take the H2O and attach one of the hydrogens to one of the monomers and the leftover OH to the other molecule. This is essential when we're trying to break down large molecules in our system. For example, imagine if we as human beings are eating, we need to digest that food. We have proteins, we have carbs, we have lipids in our food, and we have to go ahead and break them down. We have to recycle that information into our cells, utilize it as energy, or we need to naturally use them within our cells by the assistance of enzymes. And that is the purpose of hydrolysis. An example of this is if we consume sugar and water, and then we go ahead and we were to break it down, we would end up getting glucose, which also is a type of sugar, but it's a simpler version of sucrose. And then we would break it down also into fructose. This is just a small example, but overall, hydrolysis is the idea of adding in water to break covalent bonds in one's polymers to create monomers. So we are scientists, we're observing some sort of reaction, and we know water is consumed. We have molecular bonds that are broken. Because water is consumed, we know that's similar to that of hydrolysis because water comes in to go ahead and then essentially break a bond, even before I answer the choice. Now, when I'm answering the question and looking at the choices here, I notice that dehydration synthesis and polymerization is here. Even if you don't know hydrolysis and you're given the answer choice polymerization and dehydration synthesis, based on what we reviewed, we know that these are similar answers. So more than likely, you can't have two correct answers. So that's just something to note. Also, we're saying that we are creating this bond. And in this portion, we're saying we're having bonds broken. We're not creating these macromolecules. So A, D, even C, formation of a disaccharide. You don't know what disaccharide is at this moment. You know the root term di, which means two, which means we're taking one monomer and attaching it to another, especially with the term formation. So this is why I don't like answer choice C either. Given this information, I'm going to choose the best answer choice is B. Knowing that, that's the correct answer, and I hope this video helps you understand the differences in the video that we reviewed today.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the link tree. It's got all the goods for AP Bio, PSAT Math, and SAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.